Welcome, Duke alumni and friends, to this session of the Duke Alumni Forever Learning Institute, an interdisciplinary educational program organized into thematic courses. I am Tish Futrell, the Director of Academic Engagement on the Lifelong Learning team. Our unit is part of the Alumni Engagement and Development Office. Thank you so much for joining our program, Developing the Future of Medicine and Health. This program is part of our Future Forward research theme, which showcases Duke research initiatives that emphasize collaboration across campus and explores how Duke is positioning itself as a leader in innovative solutions for some of the most pressing issues of our time. Today's program is the fourth and final session in our Future Forward research series. We will host a Q&A session at the end of today's discussion. Today, we are joined by Dr. Cynthia Toth. Dr. Toth is the Joseph A.C. Wadsworth Professor of Ophthalmology at Duke University School of Medicine and a Professor of Biomedical Engineering at the Pratt School of Engineering. She is also the co-founder and consulting chief medical officer of Thea Imaging in Durham, North Carolina, where she works to bring innovative retinal imaging to the bedside for improved preterm infant eye care. In her laboratory at Duke, she studies retinal disease of preterm infants and collaborates with a multidisciplinary team to bring optical coherence tomography and into the operating microscope to guide retinal microsurgery. Whew. Welcome to the Forever Learning Institute, Dr. Toth.
Tish, thank you so much for the introduction. I'm really pleased that our group can actually come join you to share how research has certainly transformed our lives as we develop the future of medicine and health. So I am joined today by Christian Velan, PhD, who was a graduate student here at Duke in biomedical engineering. He came to us from, I think it was University of Virginia, but it could have been Virginia Tech. University of Virginia. Okay, I got it right. Right color sweater there. And he has more of a computer engineering electronics background, but came into biomedical and biophotonics and stayed on as an entrepreneurial postdoc, and I'll be introducing him soon. Shi Chen, associate professor in the Department of Ophthalmology, who is MD, PhD, came to us from Hopkins and Harvard, and Ramiro Maldonado, who did early research here at Duke, left for NIH, University of Kentucky as a leading genetic institute there, and returned to Duke to lead the Pediatric Inherited Disease group here at Duke. So I'm going to talk today about revolutionizing the diagnostic tools that we use and how we manage infant eye care, particularly for infants with diseases of the retina. This is a collaboration that involves not just the Department of Ophthalmology, but also biomedical engineering. So the three of us clinicians are ophthalmologists. We work in the eye, but we remind you that we are also neuro specialists. The eye is in fact a window to the brain. The retina, which lines the back of the eye, is actually an extension of the brain, and the visual pathway is actually more than a quarter of the brain, where we can see here the pathway going to the lateral geniculate and back to the visual cortex in the back of our skull. Within the retina, the magic occurs where light comes into our eyes, and it's in this layer here, the outer segments of the photoreceptors, that light energy is first transformed into nerve signals. It leaves this cell and starts to be processed as it goes through the many layers of the retina. And you'll see these stacked up as 1 through 7 are the major layers, and then the pigment layer below. We typically have to take an eye out. We can't go in and take little biopsies of the retina. And thus the retina has been a black box for those of us in clinical care and research. That was until 1991, when the invention of optical coherence tomography and its application to the eye occurred. We were lucky that Professor Izat of the Department of Biomedical Engineering was actually there at MIT in the period of the first development of OCT and brought his innovation here to Duke. Christian, I'm going to ask you as the engineer on the group to explain our upper diagram of what is OCT? How is it like or different from ultrasound? And how is it useful?
Yeah, so OCT can be thought of as something similar to ultrasound, except it's done with light instead of sound. So because light is has a much shorter wavelength, the corresponding resolution becomes a lot finer. So we have instead of seeing millimeter scale objects that you would see with ultrasound, we see micron scale objects that we need to see in the retina. Like ultrasound, OCT measures, in this case, the light being scattered back from the different layers of the retina. And generates a cross-sectional image, like you'd see in the bottom left, that shows you all those different brightnesses, all those different structures in the retina. These, and as Cindy will talk more about, these structures, their absence of, or any changes to them, provide doctors a strong window into the disease state or what's going on into the eye, in the eye.
Thanks, Christian. So early OCT was called time domain. The reference arm had to move around. It was a very slow technique, worked for a tabletop for someone who could position, but was not fast enough for us to capture scans, for instance, in moving kids. I tell people that OCT can be thought of like a strip of bacon. We're very lucky that the cells of the retina organize themselves in nuclear layers. Those are the photoreceptors that do the work of seeing, where my mouse is. The next layer up is where the bipolar cell and the Mueller cell nuclei are. Up here are the ganglion cells, and it's the nuclei of these cells, their axons on this surface, that exit the eye and go all the way to the lateral geniculate. How did optical coherence tomography change our world? Because we could suddenly see in the black box of the retina. Prior to that, it had been 1850 when Helmholtz showed us how to look into the eye with an ophthalmoscope. We can hold up, everyone's had that eye exam, and this is a photograph that you can take using the same ophthalmoscopic view. OCT was commercialized, put into a system. There are now over 12 different companies making commercial tabletop OCTs, and a patient can sit there and we can now see that what looked like just little yellow bumps in the retina on that left is actually a patient who has fluid under the retina, has abnormal little bumps of drusen, and we can diagnose age-related macular degeneration, yes, on the color picture on the left. But on the right, we can identify, without injecting any dye into the patient's arm, we can see where there's fluid leaking, what's going on with disease, and we can follow that fluid to see, does the patient require more treatment or can we watch and wait? This was accessible for adults, but not for young children. We here at Duke, and I in particular, had been using this in research in adult patients very early in the application of OCT. And then we would go to the NICU and look at infants and their retina, and we would use the same technology as developed by Helmholtz in 1850, modernized in 1947 when the indirect electrical ophthalmoscope went on our heads. But basically, we lacked OCT for the kids. Why does this matter? The most common blinding disease in infants is retinopathy of prematurity, abnormalities of the blood vessels that needed to develop at the 9th month of pregnancy. So we examined those kids and could not look at the retinal layers as they were developing. So we actually are proud to be the center of the universe in handheld OCT or in infant OCT imaging. The first systems were developed with Joe Izat here at Duke, and you can see this armature that went to an old style TV camera, not even a flat screen, or to a TV box. This is Ramiro Maldonado and myself at a very young age, and others who worked on the research team. Leica commercialized what developed from this device. That went off to commercialization, but at Duke, we continued to develop swept source higher speed systems, faster, lighter, and with novel engineering designs. The latter of these systems were developed by Christian Wieland. Handheld OCT has shown us unusual things such as this macular edema, these cystoid spaces. It looks like Mount Fuji in this infant. Should not be there, and no one realized it was there until we published this back in 2011. What we found in the NIH-funded baby steps over the past 9 years, imaging in the Duke nursery, is that infants who have a thinner nerve fiber layer, as seen at the bottom, have much more serious brain injury than infants who have a thicker nerve fiber layer and a healthier brain. Those infants also have lower birth weight, and when we test them for vision at age 9 months, they can't read an eye chart, but we have a special way of testing how they respond to visual stimuli. We've identified worse vision at 9 months and worse neurodevelopmental scores and worse autism risk at age 2. All of this from looking in their eye before the time that they were expected to be born. Why is this valuable? Because an MRI requires a child to leave a NICU and go into a tube, no metal attached, and be imaged. Very expensive device. OCT is a bedside tool that could be used on a daily basis to actually watch, and we have monitored how the optic nerve tissue develops in these children. Here we're using the OCT to follow an infant. Normal term delivery is at 40 weeks. So this is 7 weeks prior to the child should have been born. The child was actually born about 6 weeks prior to this. So this child we follow at 33 weeks. You can see nice vessels in the retina, still developing. Now they're starting to bulge out. That is a bad sign. We now have little nubbins coming up. We can see the stop of blood vessels along this edge. A ridge is forming, and you can see the neovascular tissue develop. But notice this is in one tiny window in the eye. OCT gives us a view of this one section, and we can now monitor before and after injection of the same drugs that are used to stop macular degeneration in adults are now used to stop the abnormal growth of blood vessels in these kids. What we're now trying to study is, do the blood vessels continue on and grow out to the periphery where they should as the child develops further? You'll notice that I'm talking about this, but Dr. Shi Chen, Professor Chen, who's on this, is actually the one who led this study. She's going to talk further about vessel development. The infrared light of OCT systems causes less stress or crying than the bright light eye exam that we use. If anyone who's had an eye exam knows, it's like a car headlight shining into your eye. The infants on OCT, you see this column of white, which is no change in their crying from before the exam, whereas infants with the indirect ophthalmoscope went anywhere from worsening by 1 to 4 levels, which is a high-pitched cry. The retinal view is also useful in identifying, is it healthy with no vascular disease, or does the child indeed have severe ROP, retinopathy of prematurity, tortuous vessels, and the thickness map shows us the bulging of the vessels out of the retina with abnormal growth of tissue. Note how the color photo dims in the periphery because of some of the restrictions of photos. Fortunately, we have been crazily successful. The startup Bioptigen was acquired by Leica Microsystems, and those systems that Joe Izett developed have been sold around the world. We published the first handbook of pediatric retinal OCT. Infant OCT is now part of the international classification for ROP. Hundreds of publications worldwide, including this one in Russian below, include this, and the OCT is in all the pediatric textbooks. It takes a village. The study that is ongoing involves Shi Chen, our pediatric ophthalmologists here, statisticians at the Perelman School of Medicine at Penn. The neonatal perinatal research unit is involved, including pediatric psychologists and neurodevelopmental testing later. Note that BME is the basis. They're at the bottom of the page because they are the core technology that allowed the study to happen. We've been invited to present this as team science to the Association for Research in Vision and Ophthalmology. This is a picture of some of the lab group, Joe Izat, who developed this. And I'm now going to move to Christian Veland. This is him receiving his hood when he moved from Mr. Veland to Dr. Veland, and his and the first trade show when we presented Thea imaging to the world. Christian, after he graduated from Duke, having been a superstar in biomedical engineering, was invited by the chair of the department he competed for one of the first entrepreneurial postdocs in Duke BME. And from there, Christian, I'll have you take it away.
Thank you, Cindy. So I guess there would be a bit of a branch in this presentation. I'll talk more about the commercial kind of entrepreneurial aspect of this, and then we'll switch back to Dr. Chen and Dr. Maldonado for some more interesting clinical stuff. So like Cindy said, after I finished my PhD, I was the Duke, one of the two entrepreneurial postdocs in the Duke Department of Biomedical Engineering. And what that allowed me to do was to take the third aim of my dissertation, which was one of the handheld OCT prototypes that was in Dr. To's lab. And it allowed us to get a company off the ground. That initial phase is kind of tough to get going. Someone has to pay the salary of the engineer at some point. It was the support of the department kind of gave us that year buffer to get this project off the ground and to work on getting some further NIH funding. So Thea Imaging consists of 3 members from Duke. I'm the CTO, so I have taken the tech that we developed at Duke, and we brought it into the commercial space. Cindy is our awesome chief medical officer, and then Hafiz Dalla is another graduate from the Duke Department of Biomedical Engineering, who's taken more of a business lean, and he is our chief executive officer. And our goal is to take what are current state-of-the-art technologies and bring them to state of practice for pediatrics. We take the high-speed, fancy OCT systems that you see in the adult clinic, take all those features out of those big scan heads and bring them into a small scan head on a portable cart that can be brought into the nursery or into an operating room and provide care for state-of-the-art care for patients who typically don't have access to OCT. And in particular, in late 2003, Leica Microsystems, the company that used to sell the Invizu the first handheld OCT, has end of life their product. So they're no longer selling handheld OCT systems on the market. So via support from the NIH, mostly through SBIRs and some funding from the NC Biotech Center, we've been able to develop a commercial prototype system that brings all these new high state-of-the-art features into the nurseries. What you can see on the right is we have this is the first version of our cart-based system. We have a small lightweight handpiece. Our handpiece weighs under 300 grams. So it's very light and nimble and easy to position above an eye. And then it's packed into a chassis on a small cart. So this is a standard medical computer cart, something that someone might put a laptop on and bring around for rounds. So this is easily portable in the NICU, easily portable to through a hospital, and can be brought bedside to bedside.
And Christian, just for context, what's the speed of the OCT and how much does it weigh compared to the current?
Right. So this system is about 10 times faster than the Bioptogen, and the probe is about 1/5 of the weight. So if you can imagine trying to image an unhappy baby, both having a lightweight, like kind of nimble handpiece and a faster scan are critically important to getting good images quickly, and then getting away from the baby and letting them get along with their day. In addition to what we've been showing as handheld OCT, that's typically done non-contact, and the non-contact OCT has a field of view of 62 degrees. While this is useful for showing the area for the macular in the center region of the fovea, which is denoted by the yellow boxes on these images, it doesn't show us enough of the periphery to be able to see the the tortuous vessels or the neovascularization that's Cindy was talking about earlier. Certainly, you can tip or you can angle the probe or potentially montage the probe, but it doesn't allow for single acquisition of the entire retina. So, in addition to the non-contact probe, T has been developing a wide field OCT lens. This provides a 146-degree visualization of the retina and allows us to capture enough of the peripheral retina that we can see the features of interest for diagnosis of ROP. One of the things we focused on is making this lens modular. So if you can see on the left side, that's a picture of the our alpha prototype. And the silver part is effectively interchangeable. So you can take a lens for, say, imaging cornea or a non-contact lens or an animal model lens and click them in and out of the receiver, and you can change the imaging profile of your system. So this is a comparison between RetCam, which is the current standard of care for imaging, wide field imaging of babies. This uses bright white light. As you know, I've done this on myself. It's super uncomfortable. And it also has problems with haze. So because light can scatter off the cornea and the iris, you can see that there is haze in the image that obscures part of it. So while this image is 130 degrees, you can see that not the entire 130-degree part is usable. There's this kind of white haze that obscures about half the image in this case. In contrast, our OCT image has a larger field of view, and because OCT uses, because of the inherent image process formation of OCT, we get to reject all that light from the cornea. So that light from that is still scattered off the cornea doesn't impact our image. So we get very clear visualization of the retina across the entire field of view. And we point out some matching vessel structures. You can see the vessel extending all the way out to the periphery. And this is where we are now. So we are working towards a pivotal study and a 510K for the system, a render of our current chassis and our system design is shown on the left, as well as the updated probe and industrial design shown on the right. So we're hoping to have this out on the market and into the doctor's hands shortly.
So pivotal study and 510K means FDA clearance for it to be a medical device available for patient care. Thanks, Christian. And it's neat to see how far you've come in the few years since leaving Duke, and especially since, if we recall, 2020, we hit 2020 and 2021, and you saw masks held in our hands to the side. We had taken them off for a moment for the picture because we were immediately, that was just ending COVID. And so to make it as a new company through that is phenomenal. So Christian, thank you. I now get to introduce Ramiro Maldonado, who has been a colleague and initially was a student that I met more than 15 years ago when he came to our lab and led the OCT effort and worked closely with Joe on one of the seminal papers on translating what to look at on the handpiece, how to operate the system, and how to use this in kids. So he was actually there for the first intraoperative OCT. You can see the handheld system is in a sterile baggie there, held over a patient in between parts of the case. The first handheld OCT, and he actually brought back the images of the first macular edema and identified it the first time in an infant in a NICU. So he was here at Duke Eye Center then for residency. He went to the National Eye Institute for 2 fellowships, one in inherited diseases, ophthalmic genetics, and one in medical retina, and then went to University of Kentucky to lead their inherited retinal disease and genetics center. He's now back here at Duke Ophthalmology, leading as part of the Duke Pediatric Retina and Optic Nerve Center, and he leads the Center for Ophthalmic Genetics. So Ramiro, it's really a pleasure to have you back as associate professor now here, and to invite you to present your work.
Well, thank you very much, Dr. Todd, and thank you all for being here. I would like to talk to you about inherited retinal diseases. And this is my passion. We are devoted to cure these diseases. But let me tell you a few interesting things about the inherited eye diseases. Number one, they are very rare. It's not infrequent that a patient, when it's diagnosed with an inherited eye disease, tries to find someone else with the same condition, that sense of belonging. And it's not uncommon that a patient would tell me, I was able to find 10 patients in the world. So they are very rare, some of them, and that's really striking to the patient. Another thing that I would like to tell you is that these conditions are explained by a defect in the DNA. So this causes the patient to ask themselves the question, why me? I didn't do anything. I was doing just fine. And now I discover that in my DNA, there's a problem that it's going to cause me trouble. It was certainly in the fate of the patient there. So that's also psychologically very troublesome, especially when parents have given this condition to their children. But bottom line, all these conditions will produce lifetime disability. So if you picture imagine here this little boy, he's not going to be able to play with his friends in the same capacity, maybe not being able to go out at night, or have a harder time learning in school. So it's really sad. In the case of adults, for example, many times they have to quit their jobs. They cannot drive anymore. They have to change their plans. And unfortunately, their fate is to go blind, and for the most part, we don't have treatments for these diseases. So it's a very challenging situation, and that's why we are so motivated to try to change this. And can Duke help? We are working on that, and I think that we're making big progress, and I would like to tell you how Duke is trying to fix these disorders. And I'm going to bring you back to the same kid and father. If we go inside their eyes, we can take a picture, and the picture that you see in the right, it's an image of the retina. Here is what we see. It's kind of like complete darkness there. This picture helps us to see the healthy retina, the healthy sensitive part of the eye, which is at the center. You see it's bright, but outside of that, it's very dark. The majority of the cells are gone. And it's not infrequent that patients come to see us in this advanced stage, And although we have better news now, we have some treatments now, we would like to diagnose patients earlier. And that's what Duke is doing, designing novel technologies for early diagnosis. Some of these technologies that Dr. Todd and Christian have talked about are these portable systems that help us to do the diagnosis early in children, in kids that are moving, that are shaking, their eyes are shaking, they cannot fixate, that it's very difficult to examine. These technologies are helping us to understand these conditions in a better way. So we are so thankful to the engineers, to Dr. Todd and her team that has brought these amazing technologies, because we are discovering significant amount of novel changes with these technologies there. If we go to the next slide, I'm going to give you a sample of the images that we are capturing in children with these inherited retinal disorders there. We can play the video on the first one. You can see the high resolution. Just think about like if someone has taken a little biopsy of the sensitive part of your eye. That is a 10 by 10 millimeter part of the retina of the patient, where we can study the cells, the blood vessels, as Dr. Chen is doing, and get so much more information about this retina. So it's fascinating, it's fantastic, and it's helping us to understand these diseases in a better way. And the good news for these patients is that gene therapy is finally here and stem cell as well. And so we have better news. We are treating patients. So we are making a lot of progress. So now that we have these novel therapies, we can advance, do a couple of clicks here because this is aerial view of the eye center. We have the research building there. If we do another click, we will see the clinical building. So we have them together, and in the middle is the operating room where we are doing gene therapy. And we can play the video here because this is showing how gene therapy is done. It's actually injected underneath the retina of the patient to deliver the correct copy of the DNA. So DNA had a mistake, remember, and now we are correcting the copy through different techniques, editing, RNA, DNA. But this is fascinating. To be honest with you, this was like science fiction. Not too long ago, 5 years ago, it was science fiction, and now it's happening. So we are so thrilled for these patients, and we are so happy that Duke is doing this surgery, Duke is doing this early diagnosis. Duke is really trying to change the situation for these patients. And not only that we're intervening in that, but we're also doing a little bit of a deeper dive into the science part. So this is a paper where we describe how the retina forms in the back of the eye in premature babies. And now we are seeing that these genetic defects can affect these developments. So this is kind of like the fun scientific part when we can track these layers. And we can only do that thanks to these amazing technologies developed at the Duke Eye Center, especially thanks to the guidance and leadership of Dr. Cynthia Todd, who has mentored so many trainees and produced all these amazing toys that we are going to call it today, but they are amazing diagnostic tools. And with that, I would like to just, share pictures of all the collaborators in the ophthalmic genetics, in the research lab, in the Darcy lab. And thank you so much for your attention to this talk.
Thank you, Ramiro, for bringing us up to date on the current status of gene therapy and how the OCT can provide not only a view of the disease status of the retina, as we talked about those cell layers where the first part of phototransduction or light converting to nerve signal, but also of the actual delivery of the therapy under the retina in surgery. With that, I'm going to introduce our last and yet another superstar speaker. So as I said, Associate Professor Shi Chen is MD, PhD, and I was going to pull up her neuroscience PhD so I could explain to you what she's done, but she's looked at the development of neuronal cells and is now looking at neurovascular development. We convinced her to stay on faculty here at Duke after her fellowship because her interest in the research lab had been in looking at the early signaling and what are the processes that allow normal development to occur. And she's particularly interested in, Christian talked about the fovea or the center of vision, where we can see 20/20. There's only one part of our retina that does that. And she is studying how all vessels develop, but particularly that area. She is an award-winning scientist. The picture there with us, I was honored to get in the picture for being known to her and working with her, but that's with the president of the Retina Society. She was receiving the Ray Margario Award, a very distinguished award. She is also a surgeon, and she is renowned for her work, particularly in pediatric retina. You can imagine the infant eye is the size of a dime. Our eye is the size of a quarter. So it's quite a difference in size if you're trying to work on a preterm infant, more of a nickel if it's a young child, very young child. And she's been interviewed and all that sort. When I met her, we were working in the world of structural OCT. So what you've heard from me and from Dr. Maldonado is talking about using OCT to see these layers that look like strips of bacon. Here's the macular edema that Dr. Maldonado had discovered. And this is Dr. Chen's information taking us further. So she took us from the macular edema, now looking out in the periphery to see actually where there's pulling on the retina that is actually splitting the child's retina apart, tearing apart the cells' connections, and abnormal growth of what we think are blood vessels. They look red on a color picture, but that we don't know much about where the blood's flowing and what's going on here. With that, I'm going to hand off to you and let you take us through the exciting research going on in your lab.
Thank you, Cindy. I'm very lucky, and it's my honor to work at Duke and be trained at Duke, and so now I do take care of adult and pediatric retina patients, and then so I also take care of babies with prematurity, and then when they have retinal dragging and sometimes they progress to retinal detachment. It sometimes is a hard thing for us to look at, and then thinking about when we were doing surgery and whether we're going to affect the rest of the baby's life. And then so I think that this is I get the opportunity, and I'm very lucky to be able to work with all the things that Dr. Toth and Dr. Wieland get to create. And so this is their THEA system, and we get to be the lucky first ones to use them in little babies in the NICU to look at how their blood vessels get into trouble. And so this is a great view, because before you can see that the only thing that we get to see on the OCT is just a small area. We can see there's a little bit pulling, but we have to orient the OCT one way versus another, and the baby is looking everywhere. We certainly wouldn't tell the baby where to look. And then now we're able to see the whole field of view, and this is going to be a game changer, I think, for babies with retinopathy prematurity, how to take care of them. And in fact, even with preliminary studies, it's already helping us even before and after surgery, or to follow something and to say that whether we should do surgery or not, or can we watch it a little bit closer. And then so sometimes it goes away, and sometimes it can avoid surgeries in a baby, which could be a big deal for the baby. And then so it's showing here, and you can see that the on the top is the color picture that Dr. Wieland was talking about, and then at the bottom is the ones from the OCT view, and even the OCT view is wider, and it can highlight where the pathology occurs in the temporal area, kind of on the left side, and on the right side that those are the areas that are having new blood vessels and they're dragging. And then so those are the areas that this baby needs treatment. So this baby ended up getting an injection in the eye to treat those little blood vessels that are abnormal. Other things that I've been working on, this is back in the days of working with Christian, and I think he generated these pictures actually when he was a graduate student. So using OCT, so the other way to use, in addition to the structure of the OCT, one way that we can use the OCT information is look at the blood flow. So this is basically looking at taking a picture at the same location multiple times, and then looking at their difference. So the only thing that flows is going to be the blood flow. And then so with that information, you can reconstruct blood vessel information and blood flow information. So that's called the OCT angiography. It's basically a non-invasive way, so you don't have to inject a dye in a baby's veins to look at how the blood vessels are doing in the back of the eye. And then so with this tool, it can generate really good pictures of the back of the eye and really detailed information about how the blood vessels are doing in a baby. So this is a baby that has retinopathy prematurity, and we can see the fine details of these developing and pathological little blood vessels. Around the center of the vision, there's these little loops, and then these little loops are pathological, and then before this picture and this imaging, we never know where these loops are located at. Are they above the retina or are they inside the retina? And then these imagings showed us that it's inside the retina. And we can also see that toward the center, the blood vessels are busier. And then so that they're actually busy developing and sending down deep branches, but they're kind of halted by this development of the disease of retinopathy prematurity, while in the periphery of the retina, the blood vessels are kind of more sparse, and then so they're not fully developed yet. So this is giving us a way to look at how the blood vessels are developing and how the blood vessels are getting into trouble in this retinopathy prematurity, which is primarily a vascular disease. And then so using the OCT angiography, if we go to the next slide, we were able to see kind of how the blood flows are in the back of the eye, and then we can see these new blood vessels. So these colored pictures with the yellow spots, and whether they're within the retina or protruding above the retina, we can see all of these now, which we were never able to see before. This is a new vascular plaque. Basically, the new blood vessels now, in addition to these little buds of new blood vessels forming, now they're forming into little plaques. And these plaques can potentially progress get into trouble and form scar tissue and lift the retina up and cause retinal detachments. And then, but with the OCT angiography, we can see the little details of these plaques. And then so we can see how the finer vessels are on the top and bigger vessels are deeper. And so these imaging give us a whole lot of new information about how the pathology of retinopathy prematurity is forming these abnormal neovascular plaques. And then then this is the movie looking at it, and then looking at the fine vessels going to the deeper vessels, we can also see there are these mysterious flat neovascularization. It's basically scar tissue and new blood vessels that are flat. And we were never able to see that before. And it's just kind of, people think on exam, but there's no real good pictures of it. And then now we can see that these are kind of a sheet of new blood vessel and scar tissue on the surface of the retina. And then they can regress, and we can follow them as they regress and see how they're doing. So those are in babies with retinopathy prematurity, and they do affect. So every NICU that they to provide a certain degree of care, they will have to have retinopathy prematurity screening. So all of the hospitals with a NICU are required to provide this service, and then so this, for the wide field OCT imaging, that is a potential application that could be applied and could potentially be a more comfortable way to screen for retinopathy prematurity. It could potentially be used by lay people and nurses rather than the shortage of pediatric ophthalmologists and pediatric retina specialists to screen for retinopathy prematurity. So I think that it does have a big market and a wide field of application. I'm really thankful for Cindy and Christian that are actually working very hard to make that a possibility. I certainly think that they're going to give us a whole lot of information that we couldn't see before. And then briefly, another thing that I'm very interested in is the fovea, the center of the vision. So you can see there's a dimple in the center of the retina, and then that dimple of the retina is the dead center of the vision there where we can see 20/20. And then so the OCT imaging can give us information about how this develops, especially because in babies, they develop in utero. And then in the NICU, in preterm infants, because the preterm infants are born early, they give us an opportunity to actually look at how the fovea develop when they were supposed to be in the womb. All of us actually have been working on how the center of the vision develop in this. And to develop this center of the vision, it is not just the neuronal structure that is gradually forming this deepening of the fovea, the center of the vision. The blood vessels also form around the fovea. So it forms what we call foveovascular zone. Basically, the blood vessels started developing around about 14, 15 weeks. So kind of end of first trimester, the blood vessels started entering the optic nerve, and they gradually spread out into the periphery of the retina. So that's the superficial blood vessel layer. And then they started to send down descending branches and then form the secondary deeper vessels in the retina. And then so OCT and OCT angiography, because we were able to do them at the bedside in the NICU and it's non-invasive, they give us an opportunity to look at how these are developing and how the center of the vision form. And then so that we can hopefully one day predict what vision the baby is going to have when they grow up. And then so these slides are talking about when babies are born premature compared to a baby that is not premature, their retina are different. So they have thicker inner retina. They also have a very small or absent foveoavascular zone. And that's kind of the blood vessels in the center of the vision that instead of missing an anastomose into this an area of no blood vessels, so that they don't interfere with the center of the vision, they actually kind of don't obey that and then form together. And then so this is also the OCT imaging can also give us information about how those are maldeveloped during development. Very quickly, going through these, and so we are able to now visualize this process, or we're working very hard to visualize this process of how the superficial blood vessel that forms, and then they form the deeper vessels, and how they anastomose into a mature fovea. And then so the OCT and OCT angiography gave us an opportunity to look at this. We can see that this is catching the blood vessel that are developing, but they haven't developed and finished developing and touching each other yet. And this is forming this foveoavascular zone. So the retina is thicker in the beginning, and then it gets thinner because it doesn't have the blood vessels in them. That we were able to, for the first time, see how a fovea is formed in the baby. So at 33 weeks, which is 2 months before they were supposed to be born to 1 month before they were supposed to be born, there was an absence of blood vessels to forming the blood vessels around the fovea. And this coincides with how the fovea forms in the center. And so then we were able to, for the first time, watch how a fovea forms in a baby, and looking at both the blood vessels and the retina part. We can also look at how persistent the blood vessels are, or the lack of development there is. So for example, this baby who received an injection in the eye to treat the retinopathy prematurity, we can see that for about 3 months, there is not much of a progression of the blood vessels developing. The fovea is still probably forming, even they're at 48 weeks, which is already 2 months after their term birth age. And it still hasn't completely formed, but gradually they do form the center of the vision. But we were able to observe this process now, and then see how the blood vessels develop in the back of the eye. Research-wise, there are different segmentation algorithm to determine how we're going to parse out this OCT information into different layers of vessels in the back of the eye. And you can see that there's we are able to parse out all 3 layers in a developing baby's eyes. And you can see the superficial layer, the intermediate layer, the deep layer, and those 3 layers that constructs the perifoveal blood vessels in the baby.
Steve, thanks for taking us in a deep dive very rapidly through some amazing research. I don't know if you realize that this is the first person on Earth who's ever looked at and identified how this is all at the same time. Previously, we could only see this view, even if we did an IV imaging in the child, that masked the deeper vessels. So we never knew that there was this large gap and the timeline of blood vessel development. So with this end of her talk, this is like what you're seeing is fresh off the research track, and I'm gonna shift so we can actually take some questions now. So thank you all. I hope you can see how enthusiastic we are about letting children have access to the same technology that we expect as adults, and how it's changing our treatment of kids with illness, but also the understanding of how the eye is developing normally. So she is understanding what timeline, what mechanisms, what things a child eats, drinks, poops, or medications they receive and how they impact that development, and how Dr. Maldonado can search for what is the abnormality that occurs in the retina relative to a gene problem. Trish, do you have questions?
Yes, there's two questions in the chat that we can pull from.
Sure.
The first question what is the market size for handheld OCTs and intra-op OCTs?
So I'm a little less familiar with intraop OCT because I haven't worked in it in a while. But if you target all the level 3 and level 4 NICUs, that's 600 or 700 NICUs in the United States. Plus, you can start adding other environments where a portable or reconfigurable OCT system might be of value, say in emergency rooms, ophthalmic operating rooms. So there are potentials for thousands of systems to be sold within the U.S. And then also by extension in other markets as well.
Great. Thank you, Christian. The next question from Bob Warren is Duke part of the ATSENA clinical trials for XLRS?
The X-linked retinasthesis trials?
Yeah, so to give you a little context, there are many inherited retinal diseases. X-linked retinoschisis is one of them. And there are gene therapies for many genes. So we don't have that particular trial here. So when we see a patient with X-linked retinoschisis, we often go through the discussion of referring that patient. But we do have several clinical trials for other genes. So it is difficult to have all the trials in one institution. So yeah, we have a good amount of gene therapy trials, but not all of them. In particular, these at SINA, we don't have it.
There's one, a great follow-up question from JN, which is when gene splicing occurs, so does the faulty eye become completely normal, somewhat improved, greatly improved? Like, what are the things like in Leber's congenital amaurosis, for example? Or
yeah, I think that the question in general is referring to what would be the expected outcome from gene therapy. And what I tell patients, or the concept in general, is that gene therapy main purpose is to stop the progression of the disease. So remember, this leads into complete blindness. So stopping the disease is very important. But I usually tell patients that any improvement would be a bonus for them because there are some cells that are still present, and with gene therapy, they can recover functionality. So in general, the concept is achieve stability, prevent progression, but there's always many cases where vision has improved.
and there's also a question, about use in retinoblastoma. Can you use OCT for visualizing?
Yes, absolutely. And actually, and then previously it's Dr. Mrityunjay, and now at Stanford, and then now Dr. Madaran and Dr. Arpita Menier. So they have shown that actually, if they don't have access to the wide field OCT at this time, but previously with the smaller field OCT, they were able to show that in the very early stages, when there's just a little bit of a change, you can't really see it on exam, but on OCT you can actually pick it up. So it is actually potentially a more sensitive way, especially within the wide field system. I think there's actually a lot to learn in terms of the internal structure, what we can visualize in the retinoblastoma, what does calcification look like? Because before we were not able to see that. And if it is a big tumor, we can't see very well into it. But I think with the wide field, it will be very exciting.
Thanks. And I think we may have to wrap up with the last question. Ramiro, is there a prospect for stem cell treatment or gene therapy for chronic retinal detachment in the future?
Well, I mean, I mentioned the stem cell because it is currently applied for retinitis pigmentosa. a phase 2 clinical trial that showed safety and efficacy, so improvement in visual acuity. So we're very excited about that. But that is for retinitis pigmentosa. I don't see how it could help with a chronic retinal detachment. Maybe you can chime in there.
Not yet. Super. Well, thank you all for the opportunity.
Great. And thank you, Drs. Toth, Chen, Maldonado, and Violin for such a great session, and to our audience for joining us today.